Hello friends, we are going to study today transcription in prokaryotes. Now when we talk about transcription, we should also get first of all our ideas between what is the basic difference between transcription and replication. Now in transcription, we are going to produce a single-stranded RNA molecule from a double-stranded DNA, whereas replication is production of new sister DNA strands. But there are certain similarities of between that is transcription and the replication. When we see the similarities, both involve the enzymes which synthesize a new strand of nucleic acid from the template strand. But in replication, the new strand is of course the DNA, whereas in transcription, the new strand is RNA. Both the process certain enzymes are required, and in transcription the main enzyme which is required is RNA polymerase. Now there is a basic difference between transcription in prokaryotes as well as transcription in eukaryotes, as there is basic difference between both the cell structure. If we see the cell structure of prokaryotes, nuclear membrane is completely absent, and that is why as soon as the DNA is transcribed to form an RNA, this RNA is immediately translated to form proteins. Whereas in eukaryotic cells, after the formation of mRNA from the double-stranded DNA, it takes a little time to come out of the nuclear membrane into the cytoplasm where it undergoes the translation. So that is the basic difference between the transcription and translation process that is carried out in prokaryotes and eukaryotes. Today we are going to see the transcription in prokaryotes. The differences between transcription and replication that are usually studied are few but of course very important. As I said, the first basic difference is due to the cell structure. The new strand that is produced in prokaryotic transcription is by addition of ribonucleic acid, whereas in replication is the addition of deoxyribonucleic acid. The second basic difference between transcription and replication is replication is having its own proofreading mechanism, or we can say DNA repair system, whereas it is totally lacking in transcription. So once a mistake made in transcription can be carried forward. So let us see now the basic processes of transcription. When we study transcription, we can study it in three phases or three steps. First is initiation of transcription, elongation of transcription, and termination of transcription. Now this slide shows you transcription, that is the whole process and the central dogma of molecular biology, which shows you that from DNA it is transcribed to form mRNA, and from RNA it is translated to form protein. So transcription is the first step in gene expression. It involves of copying gene's DNA sequence to make an RNA molecule. It is one of the steps in using information from a gene to make a protein. Now, before starting actual initiation of transcription, we should first see what is the structure of gene in prokaryotes. Now, you could see in the slides a main important enzyme, that is RNA polymerase. RNA polymerase in E. coli or in prokaryotes mainly consists of four different subunits, which is called as a core enzyme, which is made up of four units, 2 alpha, 1 beta, and beta dash. Now this whole core enzyme, when it gets attached, or we can say another factor which is called as sigma, which gets attached to the core enzyme, this whole structure is called as a holoenzyme, or the RNA polymerase is called as holoenzyme. Now it is this enzyme which is required for correct initiation of transcription, and it is this RNA polymerase which goes on addition of ribonucleotides in 5' to 3' direction. Now, in the mechanism of bacterial transcription, the Two important things that we have to study about the enzyme is of course the RNA polymerase and the sigma factor. Now, most of the E. coli promoters bound by this sigma factor are associated with RNA polymerase. Now, each of the unit is having its own specific function. You could see in the slide, alpha unit is having its own function, that is initiation and interaction with regulatory proteins. Beta is having again chain initiation and elongation. Beta dash is having a function for binding up of the DNA template strand, and the main important sigma factor is promoter recognition. There is one more enzyme subunit which is of course not shown here, but it is there, which is called as the omega. So omega is also present there within this core enzyme. So alpha, beta, beta dash, omega and sigma. So these are all various subunits of the RNA polymerase. Now, in eukaryotic RNA transcription, three enzymes are involved: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Now, prokaryotic gene structure. If you see the gene structure in prokaryotes, we could see that there is an upstream sequence and downstream sequence. Upstream sequence means the one which is on the left side of the starting point of the transcription, and downstream sequence are those sequences which are on the right side of the starting point of transcription. Now, the promoter and terminator are two important regions which are present there in the gene. It is the promoter region which is present on the upstream side, and the terminator region is present at the downstream side. Now, there are certain specific sequences in the promoter region which help the RNA polymerase to go and bind in that particular region and start the transcription. Now you could see in this diagram that we find that double-stranded DNA. So one of the strand is called as the coding strand and another strand is called as the template strand. Now it is this template strand which is used for making the single-stranded RNA. And this is the RNA polymerase holoenzyme in a whole, which goes and binds to this double-stranded DNA and starts the initiation. Now, the promoter region which is present there in the DNA is very important because it is having two specific sites which are called as -10 and -35 region. Now these two regions are very important for binding up of the RNA polymerase enzyme. The -10 region is a 6 nucleotide region which is also called as Pribnow box or Tata box because the nucleotides which are there are TA, TAAT. You could see in the figure here. The second sequence is -35 region sequence, which is 35 nucleotides away from the transcription starting point in the prokaryotic gene structure. So this whole is complete the promoter region, and it is in this region where the RNA polymerase will go and bind. Now the sigma factor binds to the promoter sequence in between -10 and -35 sequence. The core enzyme binds to the sigma factor and promoter, but DNA is still closed. Now what do you mean by closed? So during initiation of the transcription, two important parts we can say of DNA or stages we can say of DNA are observed. First is called as the closed promoter complex and second is called as the open promoter complex. The closed promoter complex is that when DNA is still untwisted, when it is in the original form. So the holoenzyme, when it goes and binds to the promoter region, it starts slowly unwinding the double strands of DNA and opening up of the closed DNA structure. So untwisted promoter region is now called as the open promoter complex. The RNA polymerase binds to -10 sequence and placed in position to start transcribing. Slowly after the starting of the transcription or initiation of the transcription, the sigma factor is released so that only the core enzyme can go forward transcribing the whole DNA. Now you could see in this structure, in this figure, first figure you could see that the sigma subunit positions the RNA polymerase at the correct initiation part, that is between -20, 10 and 35. As soon as the initiation starts, upon initiation of transcription, the sigma subunit slowly dissociates from it and the RNA polymerase moves in the forward direction. Now, the complementarity and asymmetry in RNA synthesis, only one strand of DNA is used as template for RNA synthesis. Suppose this is gene structure 1, this is gene structure 2. You could see that in this gene structure, the lower side of the DNA is called as the template strand and it is used for transcribing RNA. Whereas in this gene structure, you could see that this is the template strand and the RNA is synthesized. So any of the strand can be used. Okay. The template strand is anti-parallel to the RNA transcript. The second step in the transcription is transcription elongation. The RNA polymerase, the core enzyme, moves along to the transcribe the DNA sequence into a single stranded RNA of the coding gene. When transcribing, the RNA polymerase interact with DNA sequence forming a transcription bubble. Now bubble means in this region the whole DNA are open, unwinded. So you could see in the figure. Okay, and this is the unwinded region, this is the complete unwinded, and RNA polymerase is slowly catalyzing this reaction. For elongation or during elongation, magnesium ions also play an important role. The DNA double helix is reformed as the RNA polymerase moves forward. So as and when the polymerase enzyme goes in the forward direction, the backward structure of DNA is again wounded or it comes into an original form. Now slowly the ribonucleotides are getting added and the mRNA strand is slowly elongated. This addition of RNA ribonucleotides is of course by the RNA polymerase enzyme. The last part of transcription is termination. Now, termination requires some specific signals in the DNA template. As soon as it comes across such signals, it gets an indication that now it's time that transcription needs to stop. The termination of transcription is done by two methods. First is called as row independent termination, or we can call it as intrinsic termination, and second is called as row dependent termination. First we will see what we mean by row independent or intrinsic termination. It is also called as row independent because the termination involves the structure of termination is within the RNA transcript or the shape is within the RNA transcript. It does not require any protein from outer environment. So as RNA polymerase moves, it comes across a structure which are called as inverted repeats and it forms a hairpin stem loop structure. You could see in this figure, this is the hairpin stem loop structure. Now, this structure is having two important characteristics, a strong GC-rich stem and a loop and a sequence of uridine residues in the RNA. You could see this uridine residues. So polyuridine sites and a strong guanine cytosine rich area. So these are the two characteristic structural features of the hairpin loop structure. As soon as RNA polymerase come across such a structure within the RNA, it gets a signal for termination of transcription and transcription is terminated. The second we call it as a row independent termination. Now here we require a separate protein which is called as rho. RHO rho. It is a ring-shaped single-stranded binding ATPase protein This rho protein binds to the single-stranded RNA as it exits from polymerase enzyme complex and hydrolyzes the RNA from enzyme complex. So the rho protein does not bind to those RNA whose protein is being translated, rather it binds to the RNA after translation. So we could see here that this transcription termination is totally not intrinsic. The structure is not within the RNA, but a separate rho protein, which is ATPase protein, comes and binds to the RNA strand and it makes it free from the DNA. So thank you. This is all about transcription of RNA in prokaryotes. Thank you.